Hi everyone. Today in Metabolism of Proteins, we are here to discuss about the metabolism of tryptophan. So what is tryptophan? Just a recollection of the chemistry of tryptophan. It is an aromatic amino acid and it is also considered as a heterocyclic amino acid. And based upon the metabolic fate, it is both a glucogenic and ketogenic amino acid, and it is also an essential amino acid, that is, it cannot be synthesized in the human body. And the special group which is present in tryptophan is the indole ring. So here you need to remember that you have an imidazole ring which is present in the amino acid histidine, and indole ring is present in tryptophan. Now let us move into the metabolism of tryptophan. Okay, tryptophan is an essential amino acid, so you do not have any steps for synthesis because it needs to be supplied exclusively in the diet, whereas there are steps of breakdown, how it is broken down to reach its glucogenic and ketogenic fate. So these are the steps of tryptophan metabolism. You need not worry seeing the huge pathway and the huge structures. Just remember the names of the substrates, the products, and the enzymes which are involved in this. This pathway is otherwise referred to as kynurenine. So these are all new terms. Kynurenine anthranilic acid pathway. Okay. So this pathway of tryptophan breakdown is also referred to as kynurenine anthranilic acid pathway. These are the metabolites in this particular cycle. So if you see tryptophan is acted upon by the first enzyme tryptophan oxygenase. It is otherwise known as tryptophan pyrrolase, and it is an inducible enzyme requiring oxygen. This forms 4-mylkynurenine. So tryptophan is broken down into 4-mylkynurenine. Then you have the enzyme kynurenine formylase. So you remove the 4-myl group and you get only kynurenine. Kynurenine is only formed. Now you have hydroxylase, kynurenine hydroxylase. Hydroxykynurenine is formed. The next one is hydroxykynurenine. So first you had tryptophan, N-4-mylkynurenine, and then kynurenine, and now hydroxykynurenine. Now you have the enzyme kynureninase. This is important because you have PLP as a coenzyme. PLP is vitamin B6. Yes, so most of the enzymes in amino acid metabolism will require vitamin B6. And this is one such enzyme, kynureninase. This converts 3-hydroxykynurenine to 3-hydroxyantranilate. 3-hydroxyantranilate. Now, this is acted upon by an oxidase enzyme to be converted into few intermediates like acroleil aminofumarate. Then you have the amino cis-muconate semialdehyde, oxalocrotonate, and finally ends with ketoadipate. Okay, so these are the steps, and the intermediates, if you note, you have kynurenine and anthranilate. And here you have to note one key enzyme which is kynureninase, which is vitamin B6 dependent enzyme. Okay, so now you have broken down the tryptophan. There are no steps of synthesis. And the next comes, what are the special products derived from tryptophan? So any amino acid will have a list of products which are derived from them. And here you have 3 different special products. One is serotonin. Serotonin is another happy hormone. We already saw dopamine, which is a happy hormone, right? And that is produced from the amino acid. Do you remember? Yes, it is tyrosine. So the catecholamines, dopamine, norepinephrine, epinephrine, from tyrosine. Another happiness hormone is secreted from tryptophan. Then from serotonin, you also produce melatonin. Melatonin is a hormone produced by the pineal gland, and it helps in maintaining the circadian rhythm. It helps in maintaining the circadian rhythm. And finally, you have the niacin, that is vitamin B3. Vitamin B3 or niacin is synthesized from tryptophan. So now let us see how these special products are derived from tryptophan. The first one is synthesis of serotonin and melatonin. Okay, so tryptophan is acted upon by the enzyme tryptophan hydroxylase to form 5-hydroxytryptophan. This enzyme also I would like to mention that the coenzyme is tetrahydrobioterin. Hope you should remember this word tetrahydrobioterin. We have heard it in 2 places with relation to amino acid metabolism. phenylalanine hydroxylase, tyrosine hydroxylase, and now the third enzyme, that is tryptophan hydroxylase. These three enzymes require tetrahydrobiopterin as the coenzyme. In addition, there is also nitric oxide synthase which requires this, which we will see in the next sessions. So now this tryptophan hydroxylase will convert tryptophan into 5-hydroxytryptophan. Now you decarboxylate, that is, you remove the carbon dioxide and you form 5-hydroxytryptamine. So from an amino acid, see, 5-hydroxytryptophan is just a modification of the amino acid tryptophan. You have an additional OH group, but otherwise you have the same NH2 and COOH group. You remove the CO2, then you have only an amine group, and that is why it is called 5-hydroxytryptamine. This is the other name for serotonin. 5-hydroxytryptamine is the other name for serotonin, the happiness hormone. So here there are only two steps: tryptophan to 5-hydroxytryptophan, 5-hydroxytryptophan to serotonin. From serotonin, you synthesize melatonin, that is the hormone of the pineal gland. How is that produced? Two reactions. One is an acetylation reaction. You acetylate serotonin. Okay, you add an acetyl group to serotonin. So what is the name called? Acetyl serotonin. Okay, the compound which is formed is acetyl serotonin. After acetylating, you methylate. You transfer a methyl group, methyl transferase. Here again, I wanted you to know this particular enzyme, which I have already mentioned in catecholamine synthesis, right? This methyl transferase enzyme catalyzes a group of reactions called transmethylation reactions, and transmethylation reactions are important in methionine metabolism, and it is a frequently asked short notes also. So we already saw the conversion of norepinephrine to epinephrine. It's a transmethylation reaction. And here conversion of acetyl serotonin to melatonin is a transmethylation reaction, and this requires S-adenosyl methionine. S-adenosyl methionine. All the transmethylation reactions requires S-adenosyl methionine. Okay, so these are the simple steps of synthesis of serotonin and melatonin from tryptophan. Now let us come to the next special product, which is vitamin B3 or niacin. So how does it get synthesized? You remember the pathway, breakdown pathway, right? It was the tryptophan amino acid was broken down to 3-hydroxyanthranilic acid through a series of steps. We had many steps. I am just skipping those steps for now. It ends in 3-hydroxyanthranilic acid. From there, it can either go for the breakdown or it can go towards niacin synthesis. So there you form quinolinic acid. Quinolinic acid, which then gets converted to niacin. Here it is a phosphoribosyl transferase. Okay, so quinolinic acid, and then you go for niacin. It requires the enzyme phosphoribosyl transferase. Here I wanted you to note one particular point. You remember the steps of tryptophan to 3-hydroxyanthranilic acid. I told you to note one particular vitamin which was required there, right? Yes, it is vitamin B6. Kynureninase. That enzyme required vitamin B6, right? So here you can see vitamin B3. One vitamin synthesis requires another vitamin. Okay, so vitamin B3 or niacin synthesis requires vitamin B6 or pyridoxine. And this is a very commonly asked MCQ question. Okay. Now coming to the various disorders. We have already discussed one disorder related to tryptophan metabolism during digestion and absorption of proteins. It is Hartnup disease. We'll see that next. And before that, I wanted to mention a syndrome which is Carcinoid Syndrome. So it is a Carcinoid Syndrome and Hartnup's disease. Now let us see what is this Carcinoid Syndrome. It is a tumor of the argentifin cells of the gastrointestinal tract. Argentifin cells of the gastrointestinal tract. It is a tumor, and here this tumor starts secreting excess amounts of serotonin. Lot of serotonin is produced. So what? Excess of serotonin leads to these manifestations. Flushing, sweating, and intermittent diarrhea are the most common clinical manifestations of carcinoid syndrome, and this excess of serotonin is present. And now when this excess serotonin is present, what happens? It gets metabolized by a series of enzymes. The various enzymes break down serotonin into a compound which is 5-hydroxy-indole acetic acid. So 5-hydroxy-indole acetic acid. Okay, you have 5-hydroxy-indole acetic acid, which is a metabolite of serotonin, and it gets excreted in the urine. So you hope you remember the pheochromocytoma. It's a tumor of adrenal medulla, and vanillyl mandelic acid is a marker for pheochromocytoma. And the same way here, carcinoid tumor or carcinoid syndrome, that is tumor of the argentifin cells of the GIT. There you have the marker, which is urinary 5-hydroxy-indole acetic acid. And since here again you have excess of serotonin, so all your tryptophan goes towards synthesis of serotonin. So one more product which cannot be synthesized as a result of this shunting will be the vitamin B3 or niacin. It will not be synthesized because all the tryptophan gets shunted towards serotonin synthesis, and this leads to the development of pellagra-like features. Pellagra features diarrhea, dermatitis and dementia. If untreated, can lead to the 4th D, which is death. Okay. So carcinoid syndrome is a tumor of the argentifin cells of the gastrointestinal tract. You have symptoms of excess serotonin, flushing, sweating and intermittent diarrhea, and its metabolite 5-hydroxy-indole acetic acid is excreted in the urine, and you have pellagra-like features in carcinoid syndrome, and this is usually managed by surgical treatment. The Hartnup disease, which we have already seen, it is an autosomal recessive disorder which is because of a defective transport of neutral amino acids, and tryptophan is one of the amino acid. So as a result, tryptophan absorption is defective, and here again you have niacin deficiency and pellagra-like features which are present. Okay, and here you have another test which is done, Obermeyer test. Obermeyer test is done in Hartnup disease to detect the excretion of indole compounds. So Obermeyer test, it's a commonly asked MCQ question. Okay.